The pedagogy of learning with large classes in my practice really involves two very different but related things. The first is I feel like I have to take extra care to engage the students in the classroom. It's very easy to lose focus in a large classroom and for students to let their minds wander, I think. So I try to use all of the tricks of the trade to keep attention of students. The second thing I think is really important is to have a breakout into smaller groups. And we do that in a number of ways in my large genetics classes.
Who tends to migrate together? Total strangers? Families. Families tend to migrate together. What do families share? Genetics. They will tend to share alleles. So in the course of human history, migration, particularly when you think that most of our history, most of human history, we've existed as very small breeding groups that tend to be a little bit isolated from each other and have tended to accumulate their own particular genetic signatures over time.
In terms of student learning, the biggest challenge in a large classroom, I think, is making the student feel that I'm speaking to that student specifically, and that it's not just a mass experience where I'm just throwing information out there in the hopes that people will capture it. So I think in terms of student learning, making that individual contact is really important, whether it's eye contact and scanning the room constantly, or whether it's moving out into the classroom and speaking to people one-on-one, or just including those kinds of those techniques that make the student feel like, again, that it's an individualized experience and not a herd experience.
We have heard your feedback, and we know that you're having a lot of anxiety over Abraham Lincoln's DNA and what will be on the exam. So we've actually taken Abraham Lincoln's DNA off of the exam.
It's often difficult to gauge whether we're being successful or not in a classroom of any size, but in a large classroom particularly, I think we have to pay attention to the feedback we get from students.
What we're going to do beginning next week is we're going to allow you to do a short written assignment on Abe Lincoln's DNA. You have 5 weeks of those assignments. Each will be worth 2 points. We'll add all of those, and then that will count as a quiz in your discussion section. So we're going to keep doing Abraham Lincoln's DNA. We're going to continue to cover it in discussion, but we've taken it off the table for the exams.
The large classroom, I think, requires a higher degree of organization than the small classroom. The issues of keeping the students engaged, I think, require that you have a roadmap for your lecture and that the student has a very clear idea of where that roadmap will lead.
Take advantage of the help hours and the extra help over the weekend. It will benefit you a great deal. I wanted to remind you that tonight at 7 o'clock in the Dodd Center, Dr. Henry C. Lee is going to be giving a lecture.
In a large class, I think it's particularly important to have additional activities outside of the classroom to both engage the student in the material and to help the student better master concepts. We have 2 activities that we do in my large class. One is that we have extra scheduled help hours every week, so that the students have an opportunity to see the material in a context other than the large classroom.
For those of you who have never heard Henry Lee speak in person, I can assure you that it is a wild ride. I will also warn you that his talks are extraordinarily graphic and not for the faint of heart. But please do feel free to come to this lecture. Bring your friends with you.
Another outside activity that we use in my large class is to engage the students in attending events other than the classroom that are related to the topic under consideration. So for example, in my large human genetics class, the students had an opportunity to hear Henry Lee present a very different take on what genetics can be used for. And we provided a modest amount of extra credit for those who attended. But one of the advantages, I think, of being a student at a place like the University of Connecticut is that there are lots and lots of activities always going on that can in fact supplement what's happening in the classroom.
It's important to realize that all of us in this room have exactly the same DNA-based sequence at the vast majority of our bits of information. more than 99.99% of the DNA of each of us in this room is similar to the DNA of our neighbors. So though we may look different and we may think that these represent big genetic differences and changes, in fact, the differences between us are very subtle, very small, and very shallow.
Larger classes, by definition, have more diversity than smaller classes. In my particular case, because I'm teaching human genetics, addressing issues of diversity is not a problem because of the content of the material.
So you want to recognize that when we talk about alleles, what we're really talking about are different DNA sequences. And even though we have these different DNA sequences, we are identical at the vast majority of our information positions in our genome.
I think the most effective part of using technology in the large classroom is the ability to take advantage of all the kinds of visual media that we have.
We add another marker somewhere down the Y chromosome a little bit, and we now add another bit of information that begins to tell these two men apart on the basis of their Y chromosomes.
One of the nice things about a large classroom is that you tend to have a screen that fills an entire wall. And so the kinds of normal visuals that we would use, especially in a class like human genetics, the kinds of normal visuals that we use become incredibly imposing, and they really capture the attention. I like to say they command the student's attention rather than demanding the student's attention. And so just the sheer scope of a large classroom can be used very effectively in visual presentation.
Now remember, this is what our eyelid looked like originally. Now we've got new numbers. N has become 104, and we have 64 homozygous dominant, 32 heterozygotes, but now we have 4 plus 4 homozygous recessive who came over on the good ship AA.
As we hire more and more young faculty in our department and others, it becomes a real challenge as to how we help them be successful in the large classroom. The one bit of advice I would give to a new faculty member who maybe doesn't have much teaching experience at all, let alone in a large classroom, is to approach designing your lectures as if you were having a conversation with your students. Keep the concepts simple. Identify 2 or 3 major points that you want the student to exit the lecture with, and make sure that you focus your talk around those 2 or 3 points. Our jobs are to be beacons and to direct students to the important issues.